Hi, this is Dr. Muscatello again. In Chapter 3, we are going to cover how the immune system develops and how the cells are selected so that they can participate in an active immune response. The first thing we're going to take a look at are the different receptors on the surface of B cells and T cells. Each of the lymphoid lineage cells are identified by different cell surface markers. If you looked at those cells underneath a microscope, you wouldn't be able to tell the difference between the two, but we can differentiate them based on different molecules that they express. The B cell expresses two receptors, one that's IgM and one that's IgD, and we'll talk about the difference between those and the similarities in just a moment. The T cell receptors are alpha, beta T cell receptors, at least for about 90% of the T cell receptors, and we'll talk about that in just a minute as well. Both of these receptors are actually generated by immunoglobulin gene rearrangement, which we are about to talk about. And gene rearrangement is super helpful in the immune system because it allows us this incredible amount of diversity without having to carry way too much DNA. The B cell receptor is actually an antibody molecule, and sometimes you will also hear antibodies referred to as immunoglobulins. Because antibodies way easier to say, we are going to be referring to them mostly as antibodies. Antibodies can be found on the cell surface of B cells, as you just saw in the previous slide, or they can be secreted from plasma cells. And what we're going to do is sort of go through the functional parts of this antibody molecule because those are going to be important for questions that you may get on Step 1 for USMLE. So the first thing you want to take a look at is the structure, and notice that there are two identical heavy chains. Those are going to be the longer chains, and there are two identical light chains that come together to form the antigen binding site. The antigen binding site actually forms between the variable region of the light chain and the heavy chain and is unique among all of the different antibody molecules. Another word to describe that part of the antibody molecule is an idiotype. And there are millions of different idiotypes of antibodies that bind all of the different types of things that we can get exposed to. If you look down at the bottom part of the molecule, you can see that these are labeled as constant regions. So you can see CH1, that's the first constant region of the heavy chain, all the way down to CH3. The light chain has one constant region. The heavy chain constant region determines the isotype or the class of antibody molecule. So whether or not an antibody is IgM or IgG is determined by the heavy chain of the constant region. So a couple of really important factors. When we look at an antibody molecule, everybody wants to sort of assume that the most important part of that antibody molecule is the idiotype because it binds the antigen. And while that's obviously an important part, it's not the only important part. Because if you think about an antibody binding antigen, if it was just capable of binding antigen and couldn't really do anything else, it would have pretty much a limited use. But that's where the isotype comes in. And what's really important that you understand for USMLE is that the isotype determines the function of that antibody molecule. So not just can I bind antigen, but what can I do with it? Can I opsonize? Can I activate complement? Can I cross the placenta? All of that function is determined by the isotype or the constant region. You will also notice that there's a hinge region. The hinge region is unique to the B cell receptor versus the T cell receptor, and that's because every antibody molecule is at least bivalent, which means that it can bind at least two antigens. So given that, each antibody molecule has a hinge region that gives it a little bit of flexibility in how it can bind to that antigen. It can sort of stretch out a little bit to reach antigens since they are capable of binding at least two. So as you'll notice from the figure, there are actually two classes or isotypes of antibody that serve as the receptor for the B cell. One is an IgM and the other is an IgD. What's really important that you understand for step 1 is that these antibodies differ only in the constant region, thus their isotypes are different, but they are identical in the idiotype region where they bind antigen. This ensures antigenic specificity so that you have one cell with one specificity. If you take what we just learned about the antibody molecule and you cut off one of those arms of the Y-shaped antibody molecule and you put it into a T cell, that's essentially what the T cell receptor looks like. So it is composed of an alpha chain and a beta chain, but the beta chain is actually analogous to the heavy chain of the antibody molecule, and the alpha chain is analogous to the light chain of the antibody molecule. There is no hinge region, and there's no hinge region because T cell receptors are all monovalent. You don't have the capacity to bind two different antigens, so we don't need a hinge region. And finally, T cell receptors are sort of high maintenance. So since T cells sort of run the entire immune response, they're a little bit high maintenance. They only associate with peptides that are delivered to it in an MHC molecule. That is a super important point for USMLE step 1. That is going to work its way into lots of different questions, especially when we get to vaccinations and different types of antigens like polysaccharide antigens versus peptide antigens. So keep that in mind. We're definitely going to come back to that concept in just a little bit. So we just took a look at the B cell and the T cell receptors. Each one of those receptors is actually associated with accessory molecules within the B and T cell membrane that help transduce signals across the cell. When a lymphocyte binds to antigen, the receptors recognize that antigen, but it's these co-receptors and signaling molecules that actually are essential to transducing the signal across the cell. Those molecules are needed because if you take a look at the B cell and the T cell receptors, you see that they have very short cytoplasmic tails. In order to transduce a signal across a cell, you need sort of longer, wavier cytoplasmic tails that can associate with tyrosine kinases and be phosphorylated, and that's where these molecules actually come into play. So let's take a look at the B cell signal transduction complex. You'll recognize the B cell receptor seated in the membrane of the B cell, but also embedded in the membrane, you're going to take a look at a couple of different things. You're going to notice that the little orange sort of molecules are labeled Ig alpha and Ig beta. Those are two invariant chains that associate with every B cell receptor in the B cell membrane, and they are responsible for the signal transduction that is necessary for B cells to become activated and to proliferate. You also see next to that what is referred to as the co-receptor that includes CD19, CD21, and CD81. Functionally, the co-receptor helps to lower the threshold of antigen needed for any given immune response. We are going to talk about the co-receptor for a few other things that USMLE likes to focus on. Notice that the Ig alpha and Ig beta are composed of a heterodimer, and there are actually two, one on each side of the B cell receptor. Well, the normal function of the B cell co-receptor physiologically is to actually bind to complement and serve as a signaling molecule to help activate B cells without T cell help. USMLE really enjoys the fact that the co-receptor is an attachment point for several different types of infections. CD21 is probably the most high yield, and you really need to know that that serves as the receptor for EBV. Essentially, that is sort of really the major reason that we see a lot of the symptoms that we do in Epstein-Barr virus, is because that virus can get into any B cell regardless of its specificity. CD81 is actually the receptor for hepatitis C virus and also Plasmodium vivax. And although those are not as high yield as CD21, probably important to know as well. The T cell signal transduction complex is also known as CD3. It's way more high yield than the B cell signal transduction complex because we actually have drugs or biologics that can interfere with its function. So they make for really good questions. CD3 is a multi-chain complex that is going to associate in the T cell membrane with all T cell receptors. You can see that it is composed of several different chains, and it is definitely has the long wavy cytoplasmic tails that are needed in order to transduce a signal across the cell. CD3 is going to be responsible for that T cell being able to activate, differentiate, proliferate. So it is really an important molecule as far as the regulation of the T cell response. Therefore, we have inhibitors of that molecule that we can use, monoclonal antibodies, that can help us when we would need for the T cell response not to be as vigorous. So think about what some of those times we might need something like that might be. Maybe you might be thinking about transplants. Maybe you might be thinking about certain autoimmune diseases. So that's when we would use a molecule like that to inhibit CD3. Also, the exam likes to ask you questions about what would happen if we had a mutation in CD3. What would happen if someone developed a knockout mouse where they completely wiped out the function of CD3? And so you have to sort of think through those problems and understand that essentially the T cell wouldn't be able to function if it did not have that CD3 molecule. So now let's take a look at some of the similarities and differences between the B cell and the T cell receptor. They have approximately the same number of receptors per B cell and T cell. Each receptor can only bind one antigen. However, when we look at the B cell receptors, we recall that there can be two different isotypes per cell, at least on naive B cells, IgM and IgD, versus a T cell receptor that only has one isotype per cell. B cell receptors can be secreted from B cells when they differentiate into plasma cells. However, T cell receptors are never secreted. Because each B cell receptor is at least bivalent, they can bind two antigens versus a T cell receptor that can only bind one antigen. And that sort of plays into the mobility in that B cell receptors have to have a little bit of flexibility since they can bind two antigens, and that is the hinge region, versus T cell receptors that don't need that kind of flexibility, so they are in fact very rigid. And finally, the signal transduction molecules for the B cell receptor, we talked about Ig alpha and Ig beta, and we also talked about the co-receptor CD19, CD21, and CD81. You're going to find that CD19 is also an important CD molecule that we're going to use to help enumerate B cells within any given population or patient. And CD3 is the signal transduction molecule associated with the T cell receptor, and likewise is also important to enumerate T cells. It's important to remember that CD3 is found on all T cells, so all T helper cells and all cytotoxic T cells will have a CD3 molecule. Next, we're going to talk about how the B cell and the T cell receptors are actually generated. And this is sort of the first part of a conversation about receptor diversity. In order to generate millions of different idiotypes for B cells and T cells, we need a mechanism in place by which we can do that, because if we were trying to actually encode a gene for every single different type of pathogen that a B cell or a T cell would come in contact with, it would require way more DNA than we even have in an entire cell. This diversity is accomplished by what we call gene rearrangements, and they're pretty much identical for B cells and T cells, with a few subtle differences that we don't even really need to get into. These happen during the development of both B cells and T cells. So sort of once we get through talking about rearrangement, when you get into talking about B and T cell development, well, the hard part will already be done. Each individual inherits different segments of DNA, which we can sort of rearrange. It's just like sort of shopping with accessories, where you can have a little black dress and you can dress it up and you can dress it down depending on what you wear with it. And that's sort of how receptor diversity works. We can have the same variable regions with different D regions and different J regions, and that antigen binding site can look completely differently. We are going to talk about gene rearrangement as it happens on the heavy chain for B cells, but keep in mind this process is analogous in T cells, starting with the beta chain. Take a look at the figure on the right. You can see that the germline DNA contains multiple V segments, D segments, and J segments that will rearrange to eventually give us the variable region of the heavy chain. But also notice that downstream of those gene segments, we have multiple constant regions. So we have our C mu, C delta, C gamma 3, etc. And those are going to go into making the different constant regions of the heavy chain, which will eventually dictate what isotype we have. Now, as far as this process is concerned, USMLE is not going to get super specific on you, but we're going to go through the process, and what's really going to be important for you to focus on is sort of big picture. So, for example, what is the expected outcome if there is an issue with gene rearrangement? So, the very first process that we're going to take a look at is that we have these multiple D segments and J segments, and we are going to pick one. We're going to pick one D and one J segment from that DNA, bring them together so that we now have what we call a DJ segment. That rearrangement is accomplished by an enzyme called the recombinase, that we'll talk about in just a minute. And the recombinase is going to initiate this by rearranging that first DJ segment, and then gets to pick from any number of these different variable segments. It is going to splice out that V segment, put it into proximity with the DJ segment, and then DNA repair enzymes will come in and seal those gene segments together so that we have now what you can see here as a V, a D, and a J segment. That is going to translate into, again, the variable region of the heavy chain. So we're doing all of this work simply just to get that one little segment of the heavy chain, but that's really important because that helps us with all of the diversity that the immune system has to deal with. Now, the rest of this is just what I like to refer to as boring molecular biology. So try not to fall asleep here. What happens to DNA if we're going to try to make a protein? Well, we have to make RNA first, so we have transcription. We get a primary RNA transcript. Well, we have to sort of dress it up. We put a little hat on it, we put a little tail on it. It becomes a messenger RNA. Are you awake? Okay, just checking. And then that gets translated into protein. So what we have just done is we have made one IgM heavy chain, which we'll also sometimes called mu. Okay, and if that rearrangement goes well, then that cell is going to just continuously make that mu heavy chain, which will eventually turn into an IgM molecule. Remember that we have multiple segments, V segments, D segments, and J segments that come together to give us the variable region for the heavy chain. And remember that this process is pretty similar between B cells and T cells, and we're not going to go through it again when we talk about T cells. So just as a reminder, the rearrangement that we just talked about all occurred just to produce the variable region of the heavy chain, which you can see here colored in blue. Now, the constant regions basically comprise the rest of that molecule, which will eventually determine the function of that antibody molecule. But before a B cell comes in contact with antigen, the only constant regions that we're able to generate are IgM and IgD. And we will talk about how that happens and how you have different isotypes with the same idiotype in just a little bit. As I already mentioned, the recombinase is the enzyme complex that facilitates the rearrangement of B and T cell receptors. The recombinase is actually a multi-enzyme complex that helps to facilitate this process. Of importance for USMLE are the RAG1 and the RAG2 gene products. And the reason they like to focus on these is, of course, we can manipulate these, we can mutate these, and result in immunodeficiency diseases such as SCID. Light chain gene rearrangement is a little bit simpler than heavy chain gene rearrangement simply because there are no D segments. So we literally only have to rearrange V segments and join them to J segments. One of the differences you will see, though, if we compare light chain gene rearrangement to heavy chain gene rearrangement, is that the light chain constant regions are not all on the same chromosome. They're actually on separate chromosomes. So you can actually have kappa light chain rearrangements or lambda. They do not sort of follow sequentially like you see on the heavy chain. Also, as we just mentioned, there are no D segments. So same sort of thing happens. The recombinase comes in, helps to facilitate that rearrangement, brings in a V segment in close proximity to the J segment. The intervening DNA is spliced out, and those two gene segments are then ligated together. The rest, as we already said, is sort of just routine molecular biology. We are going to make an RNA transcript that we will eventually dress up so that it becomes a messenger RNA, which will eventually be translated into a specific light chain protein. And recall that these will either be kappa light chains or lambda light chains, unlike what we saw on the heavy chain genes, where we will always first make a mu heavy chain followed by a delta heavy chain. Just like we talked about in the heavy chains, where the beta chain of the T cell receptor is analogous to the heavy chain, the alpha chain of the T cell receptor is analogous to the light chain. Light chain gene rearrangement is responsible for just the variable region of the light chain, as you can see here, colored in light blue. The constant region of the light chain that you can see here, colored in purple, are comprised of either kappa or lambda constant regions, which in humans the ratio is approximately 2:1 kappa over lambda. In addition to the gene rearrangement that happens for B and T cell receptors, there's a whole other level of diversity that happens that we call junctional diversity. This is accomplished by an enzyme called terminal deoxynucleotide transferase, or TDT. And TDT is not sort of this type A enzyme that we're sort of used to seeing, like DNA polymerase, that sort of reads a strand and puts complementary nucleotides in, has this extensive proofreading activity. TDT is sort of the rebel, and it completely and randomly inserts bases in between the DJ and then the VDJ junctions of the heavy chain variable region. When TDT does that, repair enzymes will fill in the complementary nucleotides, and what we have done is we've altered the gene sequence, which eventually will alter the amino acids that are found within that variable region, which completely changes up the binding specificity, or at least has the potential to do so. This actually creates a pretty significant amount of diversity in the B cell and the T cell receptors. And just to note that for B cells, TDT is very active during heavy chain gene rearrangement and begins to turn off during light chain rearrangement, versus when we're talking about T cell receptor gene rearrangement, TDT is active both in the beta chain rearrangement and the alpha chain gene rearrangement. So in talking about gene rearrangement, once that rearrangement occurs, and we think about all of the things that have to sort of come together to make a functional VDJ segment, sometimes it doesn't work, right? Sometimes we're having a really bad day. Sometimes it doesn't work and can result in perhaps the production of a truncated protein or maybe a non-functional protein. Now, when this happens, you got to keep in mind there's always that second chance, because we initiate rearrangement on one set of alleles, maybe that's the maternal chromosome, and if we get a non-functional rearrangement, we can begin rearrangement on the paternal chromosome. And this all sort of fits in very nicely with when we talk about allelic exclusion, because it's nice to know that if we get rearrangement on the maternal chromosome, we shut down rearrangement on the paternal chromosome. But at the same time, if we get a non-functional rearrangement, we still have sort of a second chance. Now, if we blow the second chance, which of course we never want to do, that does not end well, and the cell will undergo apoptosis or programmed cell death, because really a B cell or a T cell has to have a functional receptor to be functional. So if it doesn't rearrange a functional protein, we go ahead and eliminate it. So we really just talked about that a cell has two chances to give us a functional heavy chain, and if that doesn't happen, the cell will undergo apoptosis. We have a very similar process that occurs with the light chain. So we have to remember, for both the B cell receptor and the T cell receptor, we have a heavy chain, or the T cell, we call it a beta chain, and we have a light chain, or in the T cell, we call it the alpha chain. Okay? And those all come together to create the receptors. Once we do get a functional receptor, the cell shuts off rearrangement, and we just mentioned this, on the other allele, and we call that allelic exclusion, right? And why do you think that that would be important, to exclude rearrangement on the other allele? Well, because when we talk about the immune system, B cells and T cells in particular, we know that there's one cell with one specificity. And what are the chances if we allowed rearrangement to happen on both the maternal and the paternal chromosome, that those would happen identical to each other? Pretty much slim to none, okay? So allelic exclusion helps us ensure that one cell, one specificity, right? So that ensures that each B cell and T cell that we make is specific for one antigen, and that helps not confuse the immune system when these cells are trying to undergo selection. We already talked about a significant level of diversity when we talk about gene rearrangements, and the fact that we can have hundreds of different V segments and several Ds and several Js that come together in any different combination to give us the variable region of the B cell or the T cell receptor. But there's so many other layers and levels of diversity that really help the immune system be able to respond to all of the different things that the immune system can respond to. So again, another level of diversity here is that any heavy chain can associate with any light chain, okay? And if we think about that, that means I can have the same heavy chain 10 times with different light chains, and that gives me different binding specificity. So that generates a completely separate an additional level of diversity for these B cell and T cell receptors. So let's summarize the different mechanisms of generating receptor diversity. We already talked about the existence of multiple gene segments for the heavy chains and the light chains for B cells and T cells, and that we have this extensive gene rearrangement where different V's can combine to different D's and J's. That gives us one really important level of diversity. We mentioned TDT and the N-nucleotide addition, which is the random inclusion of nucleotides between the V and the D and the J for B cells, and both the beta and alpha chain for T cells. What's also cool is that any heavy chain can combine with any light chain, so I can have the same heavy chain 5 times with different light chains, and that completely changes up my variable region or my idiotype and gives me a whole new level of diversity. And there's one other level of diversity that we're going to talk about in Chapter 7 called somatic hypermutation, where B cells essentially get better during the immune response. This is sort of unique to B cells, and again, we're going to talk about that after we talk about B cells in Chapter 7. So let's take a look at how this heavy chain molecule is actually completed. We've already discussed how the variable region of the heavy chain is generated through gene rearrangement, but you can see downstream of the rearranged variable region are the constant regions. The constant regions dictate the isotype or the class of antibody. The first set of constant domains that you see are mu and delta. And until a B cell comes in contact with antigens, those are the only 2 constant regions that you can generate. You will also recall that it is the IgM and the IgD molecule that are found on the surface of a naive B cell. Now, what's important to remember is that the IgM and the IgD that we're going to find on the surface of a naive B cell has the exact same binding specificity. So they're utilizing the exact same variable region. They simply just differ in the constant region. Now, the way that these constant regions are generated, keeping the same exact variable region, goes to alternative splicing. And you're not going to need to know the process here for alternative splicing, but this is something that occurs at the level of RNA rather than the gene rearrangement, which occurs at the level of DNA. So now that we've been through gene rearrangement, let's ask ourselves, how are they going to test me on this information? You don't have to know all the minute little details that we covered, but you sort of want to focus on big picture. So what kind of big picture are we talking about? Big picture in terms of what happens if gene rearrangement does not occur. And that actually should be a pretty simple question. Turns out it's not always, but if there are mutations in RAG1 or RAG2 that affect gene rearrangement, we essentially wind up with combined immunodeficiency diseases. Of course, that's a really good area for USMLE to test you on, and they can test you on it in context of a patient presents with, or they can flip it around like they like to do and test you in the context of someone's in a lab and they mutate these genes, and what do you expect to happen? So we're going to start at the bottom of this table and look at one form of severe combined immunodeficiency, which is the result of null mutations in RAG1 or RAG2, where those enzymes have no activity within the recombinase. This gives us severe combined immunodeficiency disease because if we don't generate B and T cell receptors, none of those cells get selected and we have no B cells and T cells to protect the host. So we have a complete defect in both humoral and cell-mediated immunity. And in those types of questions, personally, if I'm going to answer a question on SCID, I like to see in the question diseases that include bacterial, viral, and usually fungal. So I like to see 3 different types of pathogens involved. Not that it always has to be that way. Recombinase deficiency is not the most common form of SCID, but we'll sort of get into that when we talk about all of the immunodeficiency diseases in a later chapter. You can also have missense mutation in the RAG genes, which will result in something called Omen syndrome, which is similar to SCID, not quite as severe, but it tends to affect Th2 cells a little bit more than some of the other T cell subsets, which then results in sort of B cell inactivity. There are a lack of B cells, actually below limits of detection for Omen syndrome. And again, it's also a combined immunodeficiency because both B and T cells are affected.